Hello, everyone. Good afternoon. I hope you're all enjoying the conference so far. My name is Naomi, and I'm going to be presenting results from a project that started as my senior thesis, titled Remote Control Meiotic Drive of Sex Chromosomes. I'm going to be attempting to answer the question, can a meiotic driver act in trans? I'll give you a spoiler alert. The answer is yes, but you'll have to stay tuned to see my model, how I got to that result, and what implications it might have. Let's start with the very basics, Mendel's law of segregation. This is something we learn in college genetics or even in high school biology, and it's based on the observation that because chromosomes segregate during meiosis, the two alleles at a heterozygous locus have an equal chance of being present in a given offspring. This means that the gamete pool will have 50% of one allele and 50% of the other. But of course, no rule in biology doesn't have an exception, and the exception to Mendel's law of segregation is meiotic drive. Under meiotic drive, one of the two alleles at a heterozygous locus becomes overrepresented in the gamete pool. It can do this through a number of different mechanisms. In one set of mechanisms, it increases the success of the gametes that it's present in, for example, with R2D2 in mice. And through other mechanisms, it can harm or kill or otherwise inhibit the alternative allele, for example, with spore killer in Neurospora. So there are lots of different mechanisms of meiotic drive, and it can also take place on a variety of different genetic locations. The location that we're going to focus on here is sex chromosomes. I find sex chromosome meiotic drive especially interesting because, of course, if you have the Y chromosome or the X chromosome being passed on at a higher than equal rate, you're going to end up skewing the population sex ratio as well. Sex chromosome meiotic drive has been observed with Y drive and also created with Y drive in mosquitoes, as well as with X drive in mice. There are also countless other examples, but there's one thing that all of these examples that we have so far have in common. They're all cis drive systems. This means that the driver, the locus that is causing the skewing of these two chromosomes during meiosis, is located on the chromosome that drives. So here, for example, we have a Y drive system, and that drive locus is on the Y chromosome. Now, this might all sound very obvious. That's like saying the driver is in the driver's seat of the car. Of course it is. But is that so obvious? Couldn't it be possible to have a trans drive system where the allele causing drive is actually located on a completely different genetic element? This would be like having Y be a toy car and the autosome be a remote control that's enabling that car to drive. So to test whether this was possible, we created a population genetic model using R. And this is our setup. We have the X chromosome and the Y chromosome. You can see that I didn't mark any genes on those chromosomes because they're not relevant beyond their capacity to determine sex. And then we have an autosome with this drive locus. That locus has two different alleles. D1 causes segregation of X and Y at normal Mendelian rates, but D2 causes the spread of the Y chromosome. And it does this because when it's present in males, all of the X-bearing sperm are harmed or killed, leaving behind way more Y-bearing sperm. In the short term, this helps the Y increase in frequency, but D2 is also essentially self-sabotaging, so this is not a very effective mechanism for long-term Y drive. So to help it out, we add a second locus, which we call the assister. We model this as a sexually antagonistic locus. It also has two alleles. A1, which is beneficial from a fitness standpoint in females, which you can see from the large pink circle, and A1 is harmful in males, which you can see from the small blue circle. The alternative allele A2 works the opposite way. It's beneficial in males and harmful in females. What this locus does is it helps sustain D2's frequency in order to enable remote control Y drive. Now, let me explain the logic behind that because it doesn't sound all that simple. Let's imagine we have an autosome with two closely linked alleles, A2 and D2. These two alleles are in a kind of synergistic relationship. A2, when it's in males, experiences high fitness, so when it's linked to D2, it provides a fitness benefit to D2. D2, of course, is our driving allele, so it enables the Y chromosome to increase in frequency. And then Y feeds back around and gives A2 that fitness benefit of being in the sex in which it experiences higher fitness. And so because of this cycle, indirectly, D2 is providing a transmission benefit to A2. Another way to explain this synergy is that A2 wants to be present in a male, again, because this is where it experiences higher fitness. D2, by driving the Y, makes this a reality, and then A2 confers a fitness benefit to D2. Now, even if you're following with me and agree, okay, sure, that sounds logical, let's see if it actually works. And so to find out, we created a recursion tracking allele frequencies. And so here along the y-axis, you see allele frequency, and along the x-axis, you have number of generations. So what we see is that over time, y is able to increase in frequency. Correspondingly, the x falls down below 0.5, and D2 is able to invade, starting from mutating in to a population at which it's 0. So this confirms that remote control drive is working. But we don't yet know if it's actually following the logic that we had hypothesized. So to find that out, we also decided to track linkage disequilibrium, which is a measure of how frequently two alleles are inherited together compared to the neutral expectation. So here in this graph, we have linkage disequilibrium along the y-axis, and again, the number of generations along the x. And as we had hypothesized, we see that A2 and D2 fall into very strong linkage disequilibrium. You can see that with the orange line. A2 and Y and D2 and Y are also inherited together more likely than would be expected. In this other plot, we can track haplotype frequency, which shows us that A2, D2, Y, those three alleles, are inherited together a lot of the time. That's actually the most frequent haplotype. So these results really confirm that it is that synergy between A2 and D2 that is causing remote control Y drive. Now again, you might say, okay, cool, you must have picked some very helpful parameters to make sure that this would work. You would be right, but we went ahead and explored the rest of parameter space to see how realistic the system would actually be. So we took a look at driver strength, the strength of D2 along the x-axis, and assister strength, the strength of sexual antagonism along the y-axis. And what we see is that for the vast majority of that parameter space, we're seeing D2, represented by the colors here, rise to high frequency. You can see as it becomes more yellow, that's a higher frequency of D2. Correspondingly, we also see the population skew heavily male, as you can see with these light green colors, for a very wide region of that parameter space. Overall, what these results tell us is that sexual antagonism can indeed enable remote control drive of the Y chromosome. Now, we were really excited about this result, but it got us thinking whether sexual antagonism is the only way that this kind of system can work. We started thinking about alternative types of systems at the assister locus, and one that we came up with was a male-limited cis-acting driver. So in this case, A2 is a meiotic driver that's able to increase in frequency through spermatogenesis at the expense of A1. Through very similar logic, A2 is also helping sustain D2 frequency, which is allowing the Y to drive by remote control. I'll speed through these results because they're actually surprisingly very similar. We see again the Y chromosome increase in frequency, thanks to that linkage disequilibrium between A2 and D2, and we see that A2, D2, Y becomes the most frequent haplotype. We also see that the vast majority of parameter space is filled, so for a lot of different combinations of the strength of A2 and the strength of D2, we can see this system work. Again, the finding that we have here is that male-limited drive as well as sexual antagonism enable remote-controlled drive of the Y chromosome. Next is a little bit of a pitch to keep undergrads around in your lab, not because they will follow your instructions permanently, but precisely because they will do something entirely different. My PI told me to try to make the O chromosome drive. What he meant was to replace the XY system that we've been working with by an XO system. If you don't know, an O chromosome is actually non-existent. It just represents the lack of a second genetic element in the hemizygous sex. So here we have XO with the autosome, and it's the exact same drive system, where in any previous result, you can just replace the letter Y with the letter O. But when my PI asked me to make the O chromosome drive, I interpreted this completely differently. I thought we could start with an XY population, where males are XY and females are XX, and actually have the O come into that population, being introduced and then sustained by remote control drive. In this model, instead of shredding X-bearing sperm, D2 would just eliminate the X chromosome from the X-bearing sperm, leaving behind an O. Nothing there, so you end up with an XO male. And so what this can tell us is that we can actually simulate a Y to O evolutionary transition by using remote control drive. And so I show that this is possible with a very similar set of figures. Here we see the O chromosome rise to high frequency, the Y plummets down, and the X also reduces slightly. Similarly, this is due to linkage disequilibrium. And we can see over here even more clearly that the O is taking up a lot of that space of haplotypes, and the Y you can see along the left side is basically being eliminated from the population very rapidly. So overall, what this finding tells us is that remote control drive might be able to facilitate a transition from XY to XO sex determination. Finally, let's move on to some implications. I think this is a pretty cool finding just from a curiosity standpoint, but why should we care about this? First, there are some practical implications. For example, if this could explain an evolutionary transition, could we find any evidence for that? Could that help us explain some gaps in knowledge that we currently have about how these genetic sex determination systems can switch? Second, are there any applications for this in synthetic gene drive systems? It's becoming increasingly of interest to figure out ways to potentially eliminate, for example, malaria vectors through gene drives, synthetic gene drives, and having the ability to remote control a chromosome could end up being useful for this kind of work. Another practical implication is that currently efforts to survey and try to quantify the frequency of Y drive usually start by sequencing the Y chromosome and looking for footprints on that chromosome itself. But we might be overlooking the possibility of transdrivers located on autosomes. I also think there are some important theoretical implications. For example, in this system, is the Y chromosome actually a driver? It doesn't have any drive alleles on it, so arguably not, but does that make the autosome a driver? Maybe both, maybe neither. I definitely think that's something interesting to consider. And then finally, how does remote control drive change the notion that linked loci, enhanced drive, so those loci that are closely linked to the driver, have usually been thought to be kind of part of its team, whereas unlinked loci are usually thought to be in this kind of parliament of genes that try to suppress it. And of course, this model has the exact opposite happening. So I'll leave you with those discussion ideas, but I'd also like to acknowledge my two supervisors, Dr. Patton and Dr. Schenkel, for their help with this project, as well as a variety of funding sources. Thank you. And I'm happy to take any questions. Yes?
That was a really great talk. Thank you. I seem to notice that a lot of the time your drive is leveling out at around 2/3, seems. Do you have any explanation to why you see it often maxing out at 2/3?
The D2 allele, you mean? Or the Y chromosome?
I might have seen it a couple of times maxing out at around 2/3, around there. The Y?
Yeah, absolutely. So in this example, the Y is at about 0.55, 0.6, and that's because once you do skew population sex ratios, there's kind of a push back toward 50-50, simply because a population that's heavily skewed is going to have lower survival chances, and individuals of the rarer sex are generally thought to be more fit compared to individuals of the more common sex.
Have you tried modifying the combination parameter?
Yes, we have.
If you use a combination, it might unlink the DNA.
That's a great point, and I do actually have a result to show you. So here along the top, you can see different values for R, and you can see the further right you go, the lower the recombination and the more successful it is. And of course, if you increase recombination, here on the far left, you have 10% recombination. In that case, you do see a much less successful model.
Great talk. I was just curious why you modeled D as a sexual antagonism locus and not just a male benefit locus, because I guess the point was to stop the driver just immediately going extinct like you showed at the start.
Definitely.
So why does it need to harm females too?
It wouldn't necessarily have to. That's a great point. I think we just thought it might be more realistic. Instead of kind of giving it the ideal situations, just try to imagine a closely linked, randomly sexually antagonistic locus.
Thank you.
Yeah.
Can I ask another? If you put the D thing on the X, that might be very interesting because females get less common as it invades, and it might be self-limiting, and there's interest in making self-limited drugs.
Totally, and that's something that we do want to explore in the future. I have a little diagram here because the problem with trying to make the X drive is that the X doesn't segregate from the Y in females, so it would basically have to skip a generation and would therefore, as you pointed out, be a lot slower.
So hello everyone, my name is Juliette, and I'm going to present to you some work I've did during my PhD, and I'm gonna try to convince you that chromosomal rearrangements are a key factor of genome evolution and that we do need to do more work with them. So first, chromosomal rearrangements are mutations that can change the structure of chromosomes, so they can be inversions, duplication, deletions, translocations, and they are more large scale. They were identified very early in the history of evolutionary biology because you can see them directly on the chromosomes. You don't necessarily have to sequence data. And we know that they are present throughout the tree of life and that are responsible for some major evolutionary events. But since they are very difficult to identify with short-read sequencing data, they were often overlooked. And now that we have long-read sequencing data, more people are getting interested in chromosomal rearrangement, but we do still lack a lot of theory to try to understand them. And the few models that study them often study just a few parameters, a few impacts of chromosomal rearrangement, and not the phenomenon as a whole. And that is easily explained because chromosomal rearrangements are very difficult to understand. They are multi-scale events because they change the genetic sequence at the point of the double break strands, but they also change the genomic level of organization by changing the gene repertoire and gene synteny. They are not independent from one another because any chromosomal rearrangements can change the probabilities of future ones. So if you delete a gene, you cannot duplicate it afterwards. And as a direct consequence of that, the distribution of fitness effect is not constant and changes at every chromosomal rearrangement. That is why we believe that we need more tools to study chromosomal rearrangements because that can lead to very counterintuitive effects. But what makes them difficult to understand also makes them very difficult to simulate because they are multi-scale, they are not independent, and the distribution of fitness effect is not constant. And so they are very difficult to account for in forward-in-time models that often use a priori DFE or a fixed gene repertoire. And so we need a model in which the distribution of fitness effect is itself a product of evolution. And so that's why I would introduce you to Evol, which is a software simulation that we use in our lab, and then show you some results we have with it on chromosomal arrangement. So the key point of Evol is that we have a genomic realism. So it's bacteria inspired. We have a double-stranded circular binary sequence. And on the sequence, we can identify consensus sequences which define promoters and terminators. And so we have RNAs. And on these RNAs, we can find proteins with start and stop codons. And then we have an artificial genetic code to make sense of that proteic sequence. So the metabolism is not as realistic because we cannot model everything. So we have a mathematical metabolism. And that means that each protein is translated into a triangle function. So it's determined by its location on the functional space, its height, and its width. And these three parameters are decoded from the primary sequence of the proteins. So it's completely independent of the locus of the gene, for example. It just depends on the sequence. Then the phenotype is easily the sum of all the proteins we have. So here we just have one protein, it's easy. And then our fitness is a target fitting task. So we say that we have an ideal phenotype for the environment we are in, which is here in red. And we look at the difference between our phenotype and the ideal phenotype to derive a fitness. So that's for one individual, but of course we have a lot of individuals which are located on a grid. And we do fitness proportionate selection at that grid based on the Wright-Fisher process. So we replace the whole population at each generation. So to summarize, we have a realistic complex genome structure. We have protein functions which are independent on their locus. And we have a quite realistic but very simple population. Then we have selection, and so we do replication events, and that's where it gets interested, because we can do chromosomal rearrangement directly on the sequence. So here, for example, a duplication and an inversions. We also have local events, of course, point mutations and short indels. So here, for example, we have duplicated a gene and mutated it. So we do have a new gene on our DNA sequence, and we still don't know what the fitness effect will be at that stage. So then we can decode that genomic sequence again. We have a second protein, we have a different phenotype, a different fitness, and we can have evolution. So when we do a few loops like that, we get new genes that are recruited by duplication divergence. The protein repertoire increases over time, the phenotypes get closer and closer to the targets, and the genome can acquire a specific structure. So we can have very different genome sizes, coding proportions. We can have several genes located on the same RNAs that have similar function or not, so we can have operands. And we have an evolution that results from a mix of local mutations, chromosomal rearrangement, and selection without any a priori distribution of fitness effects. What's great with that kind of software is that we can then do impossible evolutionary experiments. So we were interested in the effect of chromosomal rearrangement, and we decided to test 4 mutational scenarios to see what different types of mutation will do to the genomic sequence. So we have one scenario with only point mutations, one scenario with all local mutations, so it is point mutations and small indels. We have only chromosomal rearrangements, which for these experiments are limited to duplication, deletions, and inversions. And we have all types of mutations at once. And we kept a constant total per base per generation event rate. And then we tested 2 different environmental conditions, far from the optimum or close to the optimum. First, if we start far from the optimum, we start from a naive individual which has just one beneficial gene, and we let it evolve for a long time with the 4 different types of mutational conditions. So here, if we look at the fitness across time, it's quite easy to see that when we don't have chromosomal rearrangement, we do way worse. So starting from naive individual, chromosomal rearrangements are far more efficient than local mutations. And that we can easily explain if we look at what happens in details in the simulations. So here on the left, you have the number of genes, and you can see that it increases very rapidly at the beginning of the simulations because we have gene duplication that are possible through rearrangements, and that's a very efficient way to improve your fitness when you are far from the optimum, much more efficient than de novo gene creation that we have with the other mutational scenarios. Having such gene duplications also hitchhikes non-coding DNA that explains the peak in genome size that you have at the beginning, but then it's downregulated and we converge to similar genome sizes. Except when we don't have chromosomal rearrangement here, you can see that the green light continues to go up, and I will go back to that later. But that's not really a surprise, right? Duplication when you have just one gene is clearly advantageous. So as to not cheat, we try to start closer to the optimum by having pre-evolved individuals who already have a full developed set of genes. And so we expect the effect of gene duplication to be strongly reduced here. So again, we try the 4 different types of mutational scenarios. And here, if we look again at fitness across time, well, having only point mutation is not great, but otherwise it's harder to distinguish the effect of our 4 mutational scenarios. And so to do that, we applied the methodology from Visa, and we fitted power laws to our fitness to try to better understand the dynamics. So it's determined by 2 parameters, A and B, B being like the initial growth rate, and A characterizing the diminishing return epistasis. So the lower the A, the more you reduce your increased rate of fitness. So you continue to increase, but you increase less faster. And what we see here is that chromosomal rearrangements are not as efficient in the short term. There is clearly a lag at the beginning for our blue curve here, but they reduce the effect of diminishing return epistasis, and so they maintain the rate of adaptation in the long term. That's just a broad view of the fitness, but then we try to look at more in details what's happening in our simulations. And so here we looked at what types of mutations are contributing to the fitness. So on the top, we have again our 4 different scenarios, mutational scenarios, and we have the total contribution to fitness for the different types of mutations. Here you can see that in our scenario, when we have all types of mutations, local mutations are still the main contributor to fitness gains. So we are not trying to say that chromosomal rearrangements do it all and that it's pointless to look at local mutations. They still are a great contributor to fitness gain, and they should be modeled alongside chromosomal rearrangement. But the big difference is that there are, here on the bottom, we have the fixation rate, the number of fixation of mutations in our different types of mutational scenarios. And we can see that the presence of chromosomal rearrangements increases the rate of fixation of local events. So it's not that themselves have a huge impact, it's that they potentiate local mutations and they allow for more beneficial local mutations to be fixed. And chromosomal rearrangements, most importantly, are very rare in the phylogeny. So they are almost never fixed here, and remind that in our mutational scenarios, all mutation types have the same spontaneous mutation rates. But as they are way more complicated and way more dangerous, because deleting half your genome is rarely a great idea, they are very deleterious and rarely fixed. So we don't observe them in real biological data as often as point mutations, naturally, but that doesn't mean that they don't have a huge impact. So, chromosomal rearrangements are ubiquitous and play a major role in adaptations, and we propose to use more simulations to try to understand their effects. We have shown that when we are far from the optimum, chromosomal rearrangements enable an effective recruitment of new genes by duplication divergence, and that can also be true for the adaptation to new environments, for example. When we are closer to the optimum, they are not so key and they are not so often fixed, so they are almost invisible in their lineage, but they increase the rate of fixation of local mutation and they help reduce the diminishing return epistasis. Finally, our results also showed briefly that chromosomal rearrangements are key to regulate genome size and the structure of chromosome, because here, as I showed you, when we don't have chromosomal rearrangements, genome size tends to increase indefinitely. And that's part of another study which I don't have to present here, but you can look at it online. And the work I presented today is already published, so you can also look at it online. So with that, I would like to thank all my collaborators and you for your attention, and I'm happy to take any questions.
I'm wondering if you've used these models to look at data from actual genomes to see if you can kind of like model something that's going on, or if there's some like connection that you made to something you see in real life.
Well, we cannot translate easily a real-life genome into a genome that we can put into our model. Because here, for example, it's binary genome, so just 0 and 1s. But we do have a version with 4 bases, so basically ATCG. And they have used it to make evolution alongside phylogenies, where we know the simulation, so we know the real phylogenetic tree. And then they can give the end sequence to phylogenetic data, to guys working with phylogenetic data, and make them reconstruct the tree and then compare to check whether which reconstruction method was more efficient. Thank you.
Okay, well, hi everyone. So today I'd like to talk about some work I've done on sexual system evolution at the University of Lausanne with John Pannell and Charles Mulon. So in flowering plants, hermaphroditism, which is defined as a situation where every individual in the population produces both male and female gametes, is ancestral and is found in more than 90% of the species. However, daiici, which is what we call separate sexes in the plant world, has evolved between 800 and 5,000 times independently, and this amounts to about 6% of the species. And what I find interesting about these repeated transitions is that in all cases, as far as I know, this has been associated with the emergence of either an XY or ZW system of sex determination, which are systems where sex is determined by a single locus, where you have one allele that encodes femaleness and the other that encodes maleness. And then you have an XY system if the male allele is dominant over the female allele, and the ZW if the female allele is dominant. And what's even weirder, in my opinion, in this system, in flowering plants, is that there appears to be a very large majority of XY systems in recently evolved dioecious species. So that brings me to the questions I would like to talk about today, which are: how do sex-determining loci evolve during transitions to dioeci, and why is XY so prevalent in these species? Right, to discuss these questions, I'm going to use a bit of theory that has a long history in evolutionary biology. It's called sex allocation theory, where a sexual system evolution is seen as a resource allocation problem, where individuals have some amount of resources that they must distribute between their female and their male function. And so a population that is hermaphroditic is a population where everybody has an intermediate sex allocation, whereas a dioecious population is a population where everybody allocates either all their resources to their female function or to their male function. And so the simplest sex allocation model goes as follows. So we've got individuals that have some amount of resources that they split between their female and male function. And this results in some fecundity given by these functions fx and mx that are called gain curves. And there's a lot of discussion about what these gain curves mean, but they are really the backbone of this theory. And I will use here just power functions for simplicity and for also historical reasons. And then following gamete production, gametes fuse randomly to produce a very large number of seeds. Adults die off, and you sample the next generation from the produced seeds. And in this very simple model, what we know is that the sexual system that is evolutionarily stable is determined by the shape of the gain curves. So if both gain curves are saturating, such that individuals accrue diminishing returns to allocating more to one particular sexual function, then hermaphroditism is favored. On the other hand, if these gain curves are accelerating, such that you have benefits to sexual specialization, then DIC is favored. But sex allocation theory sort of stops there because it's a theory that is formulated at a phenotypic level. So what I'm going to do is to try to show you what happens when you put some genetics in these kind of models. And so I'm going to start with a very simple baseline model where I assume that I have diploid individuals and their sex allocation strategy is determined by a single locus at which alleles are all additive and can encode any strategy, a sex allocation strategy between 0 and 1. And I assume small mutations, meaning that when an allele mutates, the new value that it encodes is close to the value it encoded before. And so if you simulate such a simple model, what you see in conditions that favor hermaphroditism is that the population gradually converges to some intermediate sex allocation strategy and then remains unimodally distributed around it. But if you place yourself in conditions that favor DIC, what you see is that the population will converge to this intermediate value, but then undergo evolutionary branching and the gradual emergence of a male allele and a female allele. So that at the end of the simulation, what you have is female homozygotes, male homozygotes, and then unfit heterozygotes in the middle. And that kind of situation is really a textbook situation for the evolution of dominance. So what we did next was to include dominance evolution in the model. And so the only modification here is that the locus is now made of two parts, a gene just that encodes sex allocation strategies, just like before. And then there is a promoter, a fully linked promoter that encodes the dominance relationships between the alleles. I won't go into much detail about the mechanism, but basically all you need to know is that the higher the affinity of a promoter, the more dominant the corresponding allele is. So I'm going to show a typical simulation where you have here on the x-axis the sex allocation strategy encoded by the allele, and on the y-axis the level of dominance of this allele. So each dot is an allele, and the color gives you time, forward in time in the simulation. And so what we see in a simulation under conditions that favor DIC is that the population first undergoes evolutionary branching, leading to the emergence of a male and a female allele. And then once these alleles are sufficiently diverged so that the heterozygotes become sufficiently unfit, this triggers the evolution of dominance, so that one allele becomes increasingly associated with a high affinity promoter and the other with a low affinity promoter, so that you end up with a system where one allele encodes here complete maleness and is fully dominant, and the other encodes complete femaleness and is fully recessive. That is an XY system. But if the female allele had become fully dominant and the male allele fully recessive, then we would have a ZW system. We also ran multi-locus simulations where instead of having just one locus, we have now L3 recombinant loci, just like the one I've described before, where dominance and allelic effects evolve independently at each locus. And we additionally allowed for the contribution of each locus to the phenotype to evolve. And so here I'm showing just another example simulation, where here as a function of time, you have on the top plot the contribution of each locus to the phenotype, and on the bottom plot, the sex allocation strategy of individuals in the population. And here, I first started the simulation under conditions that favor hermaphroditism. So you see that the population remains distributed for some intermediate sex allocation, and the contribution of loci drift around, but remain somewhat similar. And then I just switch the direction of selection so that now it favors DIC. And then what happens is that the contribution of all loci but one go to 0, so that this locus becomes a major effect locus that encodes all the variation in sex allocation. So that you switch from, you go gradually from a population that is hermaphroditic to a population that is dioecious. So quick summary of what I've seen so far. So in this model, what we have is that selection for DIC always leads to the evolution of an XY system or a ZW system. And that is done through the concentration of the genetic architecture of sex. And that is the emergence of one major effect locus from the initial polygenic basis and the simultaneous evolution of dominance at this locus. Right. But so concentration occurs in the model, and we see XY and ZW systems evolving, but which one is the most likely? So we ran many simulations for many combinations of shapes of the gain curves, and what we get is this. So here you have the shape of the female gain curve on the x-axis and the male gain curve on the y-axis, And you get the proportion of XY system evolving with a dark purple indicating a high proportion of XYs. And so what you see is that except in this bottom corner, which I can come back to in questions if you want to, what happens is that it appears that selection favors the dominance of the allele associated with the least accelerating gain curve. So dominance of the sex for which the benefits of sexual specialization are the weakest. And we did some math, and we were able to show that actually the selection gradient acting on dominance exactly mirrors these simulation results, meaning that we were capturing something with our math, and this allowed us to understand what was happening. So the selection on dominance is a bit complicated, but to understand it intuitively, the first thing you need to keep in mind is that selection on dominance only acts in heterozygotes. And then consider a situation where you have a male allele, a female allele, and then that are initially additive, so that you have your male homozygotes, female homozygotes, and then you have your hermaphroditic heterozygotes in the middle. Now, these heterozygotes can become more male or more female through a change in dominance, but whatever they do, they will always be less fit through female function than the female homozygotes, and through male function than the male homozygotes. So what that means is that they are stuck in a bad situation. If they become more male, they will face more intense competition from the male homozygotes, and if they become more female, they will face more intense competition from the female homozygotes. And so here, selection favors heterozygotes that make the best of a bad job. And so it favors heterozygotes that allocate more to the sex where the homozygotes are the least competitive. And that's why we get this pattern. Right. So the model I've discussed so far assumes complete outcrossing, but one thing that we know is really important in transitions to DIEC is the effect is partial selfing and inbreeding depression. In particular, because the Charlesworth have shown in the 70s that high inbreeding depression and selfing can lead to the evolution of DIEC as an inbreeding avoidance mechanism. And so what I did is that I just modified the model to allow for partial selfing and inbreeding depression. And then I calculated the gradient on dominance, and what I get is this. So here you see that the zone in which XY systems are favored is massively increased by partial selfing, indicating that if partial selfing and inbreeding depression play a role in the transition to DIEC, then we should see an excess in XY systems. And I don't think I have time to go much into the details, but the underlying reason is that selfing will tend to decrease the frequency of the male homozygotes, and so it decreases in the competition it imposes upon the heterozygotes. And so it further favors dominance of the male allele. Right, some take-home messages. So as I said, selection for DIEC, at least in this model, always leads to the concentration of the genetic architecture of sex allocation and to the emergence of an XY or ZW system. And what I like about this work is that we now have an adaptive hypothesis for the prevalence of XY in recently evolved dioecious plants. We've shown that selection favors dominance of the sex for which the benefits of sexual specialization are weakest. And so that means that any ecological mechanism that affects the benefits to specializing as a male or as a female is going to influence whether XY or ZW evolves during a transition. And one very important mechanism, such mechanism is partial selfing and inbreeding depression. And I've shown here that it appears to strongly favor XY systems. So, yeah, I'm done. Thank you for your attention. Yep.
Yep, that was really nice. So I was thinking when at the start you said about your focusing was on plants, but the kind of animal hermaphrodite groups, I think first of all, do you see a similar bias in the sex chromosome system? And then secondly, I was thinking most of these groups you can't self-fertilize in the same way that plants can. So, like, I guess from what you've explained, you'd expect then a more even sex.
So
sex
I
chromosome
don't. The thing is. So hermaphroditism is quite frequent in animals if you exclude insects, but transitions. I think most transitions between DIEC and hermaphroditism have been towards hermaphroditism rather than from hermaphroditism to DIEC. So actually we have relatively few transitions to work with. And I don't think people have looked specifically at this question because I don't think there is the same history of looking at sexual systems in the animal world compared to the plant world. So I don't know what the data looks like for these guys. And then, yeah, in animals you can have all sorts of things happening due to the fact that through sperm motility and things like that that are quite different from what we get in plants. But I mean, you could include any mechanism, like you could include population structure to have local resource and local mate competition, for example, and look at the influence of this. You could.
Yeah, I was just thinking less on there's less direct self-fertilization in some of these groups, since that seems like an important.
Yeah, in my results, yeah, it's a prior selfing thing. Yeah, but yeah, you could expect, if they are all like crossing, you could expect something a bit more even, but that really depends on the benefits of sexual specialization because here things are purely symmetrical with respect to the shape of gain curves, but you could expect that in animals one gain curve is typically more accelerating than the other. You could, if you take gain curves as a heuristic mapping resource allocation to fitness returns rather than fecundity as it's included here, then if gain curves are the same shape, yeah. But if one is more accelerating than the other, yeah. Is it time to change? Okay, thanks.
Okay, so first, thank you to everyone for coming. I'm Janna Fierst. I'm an associate professor in biological sciences at FIU in Miami, Florida. And this is work that I've been doing in collaboration with a graduate student of mine, Tori Eggers. Okay. So my PhD was actually purely conceptual analytical theory. We used to joke that it was organism-free biology. Then for my postdoc, I actually moved into bioinformatics, comparative genomics, focusing on nematode worms, particularly Caenorhabditis. And so now what I do in my work is that I actually build conceptual analytical theory, employ computational modeling, simulations to test the theory, create predictions, look at genomic data, and then try to use that as well to inform the models. And so the nematodes that we work on, I'm particularly interested in because of the reproductive diversity. And so we work on Caenorhabditis. C. elegans is the model nematode, a lot of people know. It's actually androdioecious species where there are self-fertile hermaphrodites and males in the population. And it's part of a broader species group where there are androdioecious species, dioecious species. If you step out farther, there are trioecious species with males, females, and self-fertile hermaphrodites. And there's just a ton of reproductive diversity. Within Caenorhabditis, there's at least 3 different independent origins of androdioece. And again, we have these triple gender species, and then we have really cool patterns like re-evolved sex. And so what I try to do in my modeling, in my theory, is actually take into account the real biology of the nematode system, and then also generate testable predictions. And so one thing that we talk a lot about in my group, we talk about genomes and particularly transposable elements. And so both class 1, the cut and paste transposons, and the class 2, copy and paste transposons, make up a huge portion of most eukaryotic genomes. And so transposable elements can proliferate through these different mechanisms, the copy and paste versus the cut and paste. But in general, overproliferation can actually harm the host, the organism, and it can also eventually harm the transposable element. That is, they can insert into essential genes, they can disrupt different functions. And in general, untricked proliferation is not in any organism's best interest. And so whether that's an ecological system, host virus, pathogen systems, we have sort of a core interest in how things self-regulate and attenuate. And so there's been a lot of interest in sort of recent years about external systems that can regulate transposable elements. For instance, small interfering RNAs, pi RNAs, these things. But the interesting thing with worms is that these systems have actually been lost in some groups of worms. And they also take a long time to evolve. And so again, what I'm really interested in is sort of the other side of this, and which is how can an internal regulatory logic actually regulate a system, or here, attenuate transposable element proliferation. And so what I've been modeling is actually systems of autonomous and non-autonomous transposable elements. And so a transposable element to be totally autonomous needs certain enzymes, it needs transposase, it needs certain recognition sequences for it to be able to sort of make its own transposition machinery. Non-autonomous transposons have actually been mutated so that they can't jump on their own. They're actually dependent on their autonomous counterparts for transposase and for other jumping machinery. And so what I'm doing then is actually first building a deterministic model where I've got different ways that the transposable elements interact. And so the first is that they can actually compete. And so the idea is that as the number of transposable elements increases, the genome-wide transposition rate can actually increase. And so in a competitive model, you've got both the autonomous ones and the non-autonomous ones that are continually increasing that rate of transposition. Another way that they could actually interact is that the autonomous ones could slow their transposition with an increasing number of transpositions. And so the idea with this is that autonomous transposons actually have all of their machinery in cis, and so they have a greater interest in self-attenuating, whereas the non-autonomous ones are actually competing in trans for transposase, for enzymes, for proteins, and so they kind of can't really afford to slow down. They have to keep transposing no matter what happens. And so what I do then is build a deterministic mathematical model so that we have some a priori expectations. And then I'm implementing these in slim population simulations. And so this is how I can study how our mathematical expectations then change with things like mutation rate, recombination, population size, and also mating system. And so factors that are really difficult to model in a more standard analytical scenario. And so what I find is that there's basically 4 different trajectories that the transposable elements will take. And so some of them will start in the population and just be lost very, very quickly. And so I call that blue line, the early TE loss. They can also proliferate for a while and then be lost sort of later in the simulations. And so that's that yellow line. I call that the eventual TE loss. Under certain scenarios, they can actually just proliferate without any sort of regulation to the point where they actually will either crash out of the population or the population will lose so much fitness that the population itself will crash. And so that's that sort of brownish green line, which I call the TE proliferation population crash. And the 4th one is that red trajectory. And so the idea with this is that over very, very long periods of population evolution, you can get very stable maintenance of transposable elements in the population. And so what I find is that overall, across broad ranges of parameter space, the parasitic TE regulation, where one is increasing, one is attenuating, and they have that sort of tension going back and forth, can actually maintain transposable elements at stable numbers for long, long periods of time across the entire course of the simulations. And so I can simulate it for 100,000 generations, I can simulate it for 1,000,000 generations, and the parasitic populations will maintain transposable elements. In comparison, the competitive regulation actually will lose it at different rates. And so depending on recombination rate, depending on population size, depending on mutation rate, they're actually lost slowly over the course of the simulations. And in general, what I find, which is similar to older theory too, is that transposable element maintenance requires sex. And so I started out talking about the nematode populations, how they have these androdioecious, dioecious populations. You can have some level of selfing and still have transposable element maintenance, but when you get to lower and lower outcrossing rates, when you get higher and higher selfing rates, eventually the transposable elements are lost. One thing that does happen in a lot of the populations is that they're actually able to decouple. And so the autonomous one will actually lose the non-autonomous one early in the simulations, and it's able to persist for a period of time. But then eventually, over time, it'll actually proliferate to the point where, again, it crashes out of the population. And so what that gives me then is actually some discrete predictions from the model. And so I try to structure this very discrete. If non-autonomous transposons are stable over evolutionary time and they actually outnumber the autonomous, then we're going to say that's evidence for parasitic regulation. If we find an opposite situation, then we're going to say that's evidence for competitive regulation. And so in order to test these predictions, I've been working with my graduate student, Tori, and she's actually been doing comparative genomics, analyzing nematode worm genome sequences. And so the first set of autonomous, non-autonomous transposons that we looked at was lines and signs. And so lines are long, interspersed elements. Signs are short, so they're their mutated cousins. And what we see across both the androdioecious and the dioecious worms is that the lines far outnumber the signs. And so this is about mating systems and across broad swaths of the phylogeny. And so what this gives us then is evidence for this else scenario. Autonomous TEs are far outnumbering the non-autonomous, and so we think that that is our evidence. So the second thing that we actually looked at was a DNA transposon. And for this, we looked at mutator, which is a specific family of DNA transposons that have been called mutator because they're good at mutating things. They often are associated with picking up gene fragments and moving them around genomes. And in previous analyses, we actually found discrete differences between the androdioecious and dioecious species in the mutator content. And so what Tori did was that she looked at basically 4 different species so far. C. elegans and C. briggsii are androdioecious, self-fertile. C. romanii, C. nagoni are outcrossing dioecious species. And so first she just annotated the number of mutator elements overall. And so you can see those are very high across the different populations. The second is the number of mites. And so these are miniature inverted transposable elements, very, very short little non-autonomous sequences, and then also mules. And so mules are mutator-like elements. And then she went in and tried to do these even more specifically. So mutator mites are mites that we can specifically say came from a mutator because of their sequence identity. And then she looked as well for transposase genes. And again, they have to have an active transposase gene in order to be fully autonomous and able to transpose. And what she found was that there's actually this huge variation in this pattern for the mutator or the type 1 DNA transposons, where you have a huge number of non-autonomous sequences and very, very few that can actually be competent autonomous sequences with that transposase gene. And so the reason for this is because the TEs actually have this extremely messy history. And so this is graphing out the length and nucleotides for mutator, the mites, and mules. And what should actually be here for a fully autonomous, competent transposon for mutators should be more like 2,000, 3,000 nucleotides. And so what's happening is that even these mutator fragments are actually mutated bits that are a history of transposition. And so this, for me, doing theory is always, I think, why it's important to stay in contact with biological data, is because when we talk about cut and paste transposons, it turns out they're not cutting along the lines. They actually have this phenomenally messy history of transposition, and so that's what we see in our data, and that's what we see in genomes as well. And so this is basically where we are. We have this sort of conflicting evidence. And so I will conclude this portion. So conclusions from theory is that we have this sort of clean theory that predicts that our regulatory logic can very deterministically say if it's going to be a stable or unstable pattern for transposable elements. But when we actually look in the genomes, what we find is this terrifically messy history. And so it's a history of transposition that's also being coupled up with active autonomous and non-autonomous elements. And so what we're concluding then is that it looks like some of the transposable element families are operating under these competitive interactions, whereas others are operating under these parasitic interactions. And I just wanted to also acknowledge funding sources. If you're hearing more about this, Tori's actually talking Tuesday at 12 about doing a deeper dive into transposable elements and worm genomes. I have a postdoc, Rohit, who's talking more about worms and mutations in a broader context. And then also Danny Morales is talking more about transposable elements in the context of mosquitoes. And a final thing, just wanted to say that I'm searching for graduate students and postdocs. If you're interested, please get in touch. We have lots of projects going on, worms, flies, viruses. We just got a new project funded where we're actually exploring some of these same ideas about self-regulation and attenuation, but in a host pathogen system in a fly and a virus. And we have another project where we're thinking about mutations as structural variants, so transposable elements, gene duplications, gene deletions broadly, and how we can model those as well. And with that, I'll take any questions. No, not at a broad level, although we don't have a very good sample of sort of really well-assembled genomes that we can use to study transposable elements. We did a comparative analysis like a year ago, and we found that there's not a really good clear pattern. Every time you think there's a pattern, there's like one or two outliers, and so we need more samples, better genomes. The one that did fall out as correlated with the genome size differences was that mutator one. And so that's one of the reasons why we're studying that one as well.
Okay, thank you all for your patience. Thank you to our saviors. Okay, I'll try and make sure we get wrapped up. Thank you all for coming. It's the last session of the day. So my name's Tom, and I'm currently based at IFEM's Riken in Japan. And so I'm going to be talking a little bit about the evolutionary genetics of paternal genome elimination. Before I begin, I just want to thank some of the people involved, in particular Rob, Andy, and Laura, who've been involved both in this project and some empirical work we've been doing trying to test the predictions that we've generated from these models. So I'm first going to talk a little bit about the background to these systems, and then give some results about expectations about genetic diversity within populations, and then about genetic divergence between populations. So three groups of kind of interest that I'm going to be talking about today are the fungus gnats, the gormages, and the globular springtails. So these three groups are really species rich. They comprise tens of thousands, if not millions of species in some of these groups. And they're ecologically incredibly diverse. They include pests, but also pollinators, and they span from the Arctic down to the tropics. But what unites these three very diverse groups is they share the same non-standard genetics. So these three groups exhibit both paternal genome elimination, but they also have X chromosomes. So if you went out into the wild and collected some individuals, what you'd see is something like this, where the males are diploid. They have autosomes from both their mothers and from their fathers, and they have a single X chromosome which they inherited from their mothers. And the females, again, look fairly normal. They have two X chromosomes and two autosomes, one set from each parent. But something a little bit strange happens when they undergo meiosis. In particular, the males have a non-standard meiosis where they eliminate their paternal origin copies of their autosomes, so that when they produce their sperm always contain their maternal origin autosomes as well as their maternal origin X chromosome. The females are fairly standard, so they pass on a kind of combination of their maternal and paternal autosomes and X chromosomes. So what you can see is this will mean that all the zygotes that are produced actually are diploid for the X chromosome. They contain X chromosome, they're XX. And then actually the embryos that will be destined to become males early in development will eliminate always eliminate their paternal X chromosome. So what this means is that while both sexes get genes from both parents, by eliminating the paternal origin copies in males, what this means is that the autosomes in these systems and the X chromosomes exhibit the same transmission genetics, but they have different somatic genetics. So what are some of the population genetic consequences that might arise from this system? So firstly, if we think about some kind of classic popgen parameters, first of all, if we think about effective population size, in a kind of standard system, what we'll see is that the autosomes and the X chromosomes have a different effective population size, and the X chromosome will typically have an effective population size 3/4 that of the autosomes. However, in our PGE systems, right, the X chromosome and the autosomes have identical transmission genetics, and that's what's determining the effective population size. So actually, the X chromosomes and autosomes in PGE systems will have an identical effective population size. Again, if we think about kind of selection coefficients in these systems and the effective selection coefficient, again, in a standard autosomal system, genes will spend half their time in females and half their time in males. In both systems, you'll be diploid, and so in both situations, the selection coefficients will be modulated by the dominance coefficient. In our X chromosomes, they spend 2/3 of their time in females and 1/3 of their time in the haploid males, and in that third of the time, they won't have dominance modulating the selection coefficients. But for our PGE autosomes, actually, it can be some kind of combination of the two. They're going to spend 2/3 of their time in females and 1/3 of their time in males, but in both situations, they're going to be diploid, and so in both situations, the dominance coefficient is going to modulate the selection coefficients. And finally, we can think about the mutation rate. So again, if we think about the kind of effective mutation rate we're experiencing in the genome, in an autosomal system, half the time in females, half the time in males, but in our PGE systems, for both the autosomes and X chromosomes, the mutation rate in females will be relatively more important because we spend more time in there. Okay, so let's think about now how that might affect some things we might go out and measure. So genetic diversity, of course, very important and might be driven by the balance between different kinds of processes. So in particular, I'm going to think about mutation drift balance and mutation selection balance. So if we think about the balance between mutation and drift, we can imagine a process mutations are entering our population and they're kind of lost or fixed with some frequency by drift. And the balance between these processes will give us kind of pi, our pairwise genetic diversity, and we can write that out as simply 4 times the effective population size times the effective mutation rate. So if we again think about our PGE systems, what are we seeing? They're going to have the same effective population size, the same effective mutation rate. So regardless of aspects of the demography, whether it's some kind of sex bias or differences in the mutation rate, we'd expect them to have the same neutral genetic diversity. And that's what you can see on the plot on the right-hand side here. On the kind of y-axis, I have the relative genetic diversity on X chromosomes and autosomes, and on the bottom we have kind of male reproductive skew. So if we think about our PG systems, again, regardless of the amount of skew, it should be equal. And that's not like our standard systems where factors such as the demography will modulate the relative genetic diversity we expect to see. Of course, another process that might maintain genetic diversity is the balance between mutation and selection. So let's imagine a situation where deleterious mutations are entering our population and selection is purging them with some frequency. So the kind of equilibrium allele frequency we expect to see for our deleterious allele is going to be approximated by the effective mutation rate and the effective over the effective selection coefficient. So again, if we think about our PG systems, they can have an identical mutation rate, but selection's always going to be more efficient on the X chromosome because we're kind of haploid in those males. So what this is going to mean is that as mutations become increasingly recessive, we're going to see an increasing discrepancy between the frequency of the deleterious allele on the autosomes versus the X chromosomes. And actually, because in this situation, the PG autosome and the standard autosome will behave kind of identically. So we can now think about what are some of the consequences for genetic divergence as well between populations. So again, there might be a number of processes that drive differences in the kind of fixation of mutations between populations, and I'm going to focus on neutral variants and beneficial variants today. So if we think about our kind of neutral substitution rate, you can imagine a process where mutations are entering our population, and then with some frequency, with some probability, they're going to fix in our population. And so our substitution rate, K, is going to be given by the number of new mutations that are entering in a particular class. That might be new mutations in males or females. And then in that class, there's going to be some probability of fixation. And when we kind of sum across our different classes, the kind of substitution rate we're going to get is just given by the effective mutation rate, mu. So again, as we explored earlier, the effective mutation rate will always be the same on our autosomes and our X chromosomes under our PGE systems. So what we're going to see is that regardless of any kind of sex bias in the mutation rate, we'd always expect to see an equivalent neutral substitution rate on our autosomes as compared to our X chromosomes. So again, we can see that on our plot on the right-hand side here. I've imagined there's some kind of male mutation bias. This is, quite common. And in our PGE systems, that shouldn't affect the relative substitution rate. That's unlike, again, a standard system where the male mutation bias will affect the relative substitution rate for our neutral alleles. Okay, well, what about beneficial variants? So again, we can imagine a similar process. Beneficial variants are entering our population, and there's some probability that they get fixed in the population. So again, we can model this, and we can calculate the fixation probability. And if we imagine that the mutation is relatively weak, then we can approximate it by this expression here. So again, it's 4 times the effective population size times the effective mutation rate and the effective selection coefficient. So again, if we think about our PGE autosomes and X chromosomes, what are we going to see? Again, the same effective mutation rate, the same effective population size. The only difference is going to be driven by the effective selection coefficient. And as we saw earlier, that's always going to be less on our autosomes than our X chromosomes. So that's what we can see on our right-hand side here. Regardless of the degree of dominance, our kind of dotted line here is always going to be less than 1. We'd always expect a faster substitution rate for adaptive substitution rate on our X chromosomes than our autosomes under PGE. And again, that's not like the standard system where normally we only get this effect when mutations are on average recessive, and actually when mutations are on average dominant, the autosomes are expected to adapt faster. However, this assumes that adaptation is limited by mutations entering our population. What instead might happen if adaptive variants are drawn from currently standing genetic variation? So we can imagine a process where maybe initially the mutation is deleterious in our population, is being held at some frequency. The environment changes and that variant now becomes beneficial. And what we're going to see is that now if we calculate what's the probability that at least one of those copies initially in the population, because we're going to be at multiple copies, will go to fixation, and we can calculate that fixation probability. And we can roughly approximate this on the right-hand side with kind of 4 times the effective population size and the mutation rate. And then what we have is the relative selection coefficient during the beneficial phase over the selection coefficient in the deleterious phase. So if we run through this, what we're then going to see is actually the dominance in the 2 phases, if we assume equal dominance, is going to cancel out. And so actually the only difference we're going to see is going to be driven by this kind of term on the left. And that's going to be identical for our PGE autosomes and X chromosomes. So actually, if this is the kind of process that adaptation is occurring, expect it to be equal on our autosomes and X chromosomes under PGE, and this will be less than a typical autosomal system. Of course, that makes a very specific assumption about how dominance is between the beneficial and deleterious phase. So what if we kind of consider some alternative assumptions? So one common situation might be reversals of dominance, where maybe mutations during the deleterious phase are typically recessive, but then the beneficial variant is typically dominant. And if this is the case, what we actually see is now that the autosomes can adapt much faster under PG than the X chromosome, because this deleterious variant is now being held at a higher frequency, so when it becomes adaptive, there are many more copies, and so there's a much higher probability it initially fixes in the population. Okay. Not kept you too much longer. Okay, so kind of in summary, what we can see is that what's really nice about this system is that because of the identical transmission genetics, all the kind of neutral processes that we might consider are always going to be identical, regardless of any complexities in the demography or mutation rates. And that might be the substitution rate or the genetic diversity. In contrast, when we think about selective processes, whether that's kind of deleterious variants or beneficial variants, these are going to be now uncoupled in the autosomes and our X chromosomes in these systems. And so they're a really kind of powerful kind of set of experimental systems with which to try and isolate the effects of haploid selection. Okay, and so yeah, thank you all for listening. And if you want to ask me any questions, then we can do now. Or if not, I'm also presenting a poster at board H18, so if you want to hear about chimerism and altruism, then yeah, you can come talk to me there too. So thank you. That's a good question. So it really so the ones we're interested in, these three groups, the springtails, the gall midges, and the fungus gnats, yeah, they're expressed. There are PG more generally is very diverse, and so there are species, lots of PG species not included here, and that's more heterogeneous. In some, yeah, the paternal origin copy is silenced, but for these three, it seems like they're expressed. Yeah.
So it seems that in males, the whole genome really has less effective selection. So do you expect to see some alleles that are beneficial in females but then deleterious in males?
Yeah, so yeah, I haven't focused here on all the sex-specific components. What if we're considering male-limited expression or female-limited expression? Right, so one thing is sexual antagonism, right? So normally with sexually antagonistic alleles, in the simplest case, kind of you've got spending more time in the females, less time in the males, but the males are haploid, and actually if it's additivity, those two things balance. What's nice about these PG systems, right, is that's not true. You're spending more time in the females, but you've got equal fitness effects in the two sexes. So actually, on average, the kind of autosomes in these PG systems are much more conducive to female beneficial variants invading. It's much easier for anything female biased to invade on the autosomes, that is. Yeah. Does that answer the question?
Yeah,
Okay.
It's really interesting as well because they don't have a male-specific chromosome. So
Yeah.
they don't have anything to maybe fight back against the sexually antagonistic alleles.
Yeah, exactly. Yeah. Okay. Yeah.
What are the implications of these differences in population genetic processes as opposed to classical sex let's say, in terms of the fixation of these systems?
Sorry, do you mean in terms of the evolution to these systems? Yeah, I haven't got a good answer for you. Yeah. Yeah, if you want to ask about why PG evolves, you should speak to Andy's probably the best. Let's see, he'll know a bit more than me. Yeah. Cool. Well, yeah, thank you all for coming. Thank you for being patient.